In this video, we are going to talk about the neuromuscular junction. So first off, the neurons that stimulate skeletal muscle fibers to contract are called somatic motor neurons. Each somatic motor neuron has this axon, a thread-like axon, that extends from the brain or spinal cord to a group of skeletal muscle fibers. Muscle action potentials arise from the neuromuscular junction, abbreviated NMJ, and this is the synapse between the somatic motor neuron and the skeletal muscle fiber. So the neuromuscular junction is actually the junction between the somatic motor neuron and the muscles themselves. So we have this new membrane protein called a voltage-gated channel. So a voltage-gated channel is actually a channel that opens when it reaches a certain voltage. When an action potential reaches it and it reaches a certain voltage, it will open. So a voltage-gated calcium channel is a channel that when a certain voltage is reached, it will open and it allows only calcium through. So at the neuromuscular junction, when we have an action potential that reaches the end of the neuron, it will cause this influx of calcium. And when we have that influx of calcium at the end of the axon of the neuron, it exocytoses a neurotransmitter into the cleft, or the space between the neuron and the muscle. These neurotransmitters will then bind to ligand-gated sodium channels. So these are channels that open when a certain ion or molecule binds to them. Ions and molecules are otherwise known as ligands, so they're ligand-gated. And this causes that influx of sodium into the muscle itself. Once the sodium enters into the muscle, it depolarizes, it changes voltage, and then it causes this release of calcium from the sarcoplasmic reticulum and starting that excitation coupling reaction. The excitation contraction coupling. Now, once the neurotransmitter has bound to the respective membrane protein, it's actually broken down. And then without this series of events of the action potential coming from the neuron, crossing that cleft and binding the neurotransmitters to the muscle, we couldn't have this muscle contraction. So we'll go over those in the next couple slides. So first off, we're going to talk about the definition of a synapse. So first, a synapse is a region where communication occurs between two neurons or between a neuron and a target cell, in this case, a muscle fiber. So a synapse always includes the neuron axon end bulb. So here we have this neuron axon end bulb. It also includes the space between the two communicating cells, and it also includes whatever cell that the neuron is communicating with. Sometimes another neuron, in this case a muscle cell. So a synapse is going to be all three of those, the space and then the pre and post synapse. At most synapses, we do have this small gap. This is called a synaptic cleft. It's labeled right here. This is the space between the two cells. It separates the two cells. Because these cells don't physically touch, the action potential cannot just bridge that gap from one cell to another. Instead, we have the first cell actually communicates with the second cell by means of a chemical messenger, which is called a neurotransmitter. We did briefly talk about this in that overview in the last slide, but this neurotransmitter is able to bridge that gap and go from one cell to the other across that synaptic cleft. So at the neuromuscular junction, You have the end of the motor neuron, which is right here. This is the end of the motor neuron, the end of the axon, and this is called the axon terminal. As you can see, it's labeled right here. This is just the axon terminal. It's the end of the axon, or the terminal end of the axon. And this divides into a cluster of synaptic bulbs. So here we have one synaptic end bulb. The bulb is just the end bit of the axon. You can see here this bulb. And within each bulb, we have hundreds of these membrane enclosed sacs called synaptic vesicles. And you can see them, I think, oh yeah, it is labeled here, but you can see them right here, these little circles here with these neurotransmitters within them, these vesicles with neurotransmitters in them. And there are several different types of neurotransmitters that are used at different synapses, but between a neuron and a muscle cell, they're always going to be acetylcholine. And usually this is abbreviated ACH. Acetylcholine. So that's always going to be the neurotransmitter that's used between a somatic motor neuron and a muscle cell. Now the region of the sarcolemma that's opposite the synaptic end bulb is called the motor end plate. So this is the part of the muscle cell where we are going to have that connection with the motor neuron. So it's called the motor end plate. And this is the muscle part of the neuromuscular junction. Now within each motor end plate, we have like 30 to 40 million acetylcholine receptors. You can see them here with these little green membrane proteins here. These are going to be acetylcholine receptors. This is an integral membrane protein, and it's very specific to bind to acetylcholine. So here we can briefly look at the steps of this neuromuscular junction that happens when we have this action potential or nerve impulse that comes down the motor neuron. And remember, we have these voltage gated calcium channels here that when the voltage changes, they open and they allow calcium through here, and that calcium actually allows these vesicles with the acetylcholine to actually bind to the end of the synaptic end bulb and exocytose that acetylcholine into the synaptic cleft. So now we have a lot of acetylcholine in this cleft here, and then we can go into this picture here. As you can see here, this acetylcholine is exocytosed into the cleft, released from those vesicles. And then we have the acetylcholine will actually go to the motor end plate on the muscle and will bind to these acetylcholine receptors. And once these acetylcholine receptors have acetylcholine bound to them, they open up and they allow sodium through. So once the sodium goes through, it actually triggers a muscle action potential. So we have that muscle action potential is produced, and then we go into that excitation contraction coupling, because we have that muscle action potential that subsequently will release calcium from the SRs in the muscle. Remember, we went through that whole contraction cycle, and that happens in the muscle down the road here. But then what happens to that acetylcholine once it binds to the receptors? So you can see here it binds, it allows sodium to go through into the muscle, but then it's released. And it's released here, and then we have to break down that acetylcholine, because if the acetylcholine isn't broken down, it will continue to bind, causing more action potentials. It'll be released, and then if it's not broken down, it can bind again and again and again, causing more and more action potentials and causing your muscles to contract and contract and never to be relaxed, because that acetylcholine keeps on triggering muscle action potentials. So because of that, we have to have these processes in place to actually break down the acetylcholine once it's used. So here is actually what a neuromuscular junction looks like from an electron micrograph. So here you can see the neuron here, this somatic motor neuron, just labeled there. It's going to be this big lighter colored thing. And you can actually see these muscle fibers here. They're actually striated, you could see. And then you can also see this neuromuscular junction right here. So this neuromuscular junction has the synaptic end bulbs here, and they're going to be a neuromuscular junction at each of these synaptic end bulbs, and each of these motor end plates are going to be on the other side of those synaptic end bulbs. Okay, so we have a nerve impulse or a nerve action potential actually causes a muscle action potential with these following steps that we're going to go through. So in the first step here, we have the arrival of this nerve action potential here, and this is at the synaptic end bulbs, and it stimulates those voltage-gated channels to open. So are going to be the voltage-gated channels on the nerve synaptic end bulb here. And this causes those voltage-gated channels to allow calcium into the synaptic end bulb. Because calcium ions are in more higher concentration in the extracellular fluid, you actually have calcium ions that will flow inward through the open channels into the presynaptic membrane, and then these calcium ions stimulate the synaptic vesicles, remember, which contain acetylcholine, to actually undergo exocytosis. And so these little red dots here are going to be the acetylcholine. I'm going to use a different color. So here we have the acetylcholine in these vesicles here, and they are released, as you can see here, by exocytosis. And they will enter that synaptic cleft, travel across the cleft between the motor neuron and the motor end plate. So here you can see this. That's going to be step 1. So we're going to have triggering the release of acetylcholine. At step number 2, this is going to be the activation of acetylcholine receptors. So each acetylcholine receptor actually requires 2 molecules of acetylcholine to bind to it to open. So once those 2 molecules of acetylcholine bind to the receptor on the motor end plate, it will open those ion channels that allow sodium to cross the membrane into the muscle. So here we have sodium that will actually enter into the muscle, and it triggers an action potential within the muscle. So that's actually going to be step 3. We have that inflow of sodium down its electrochemical gradient, and it makes the inside of the muscle more positively charged. And that change in the muscle potential triggers a muscle action potential. I'm going to briefly go over action potentials, but we will go into it in more detail a little bit later. But just very briefly here, this is going to be the inside of a muscle cell. Sodium is going to be in a lot higher concentration in the extracellular matrix than inside the cell. So on the inside of a muscle, this is just going to be my muscle. We have a lot of sodium on the outside. And quick little diagram here. Most cells have a resting voltage of about 70 millivolts. That's just their resting membrane potential, and that has to do with the amount of ions on either side of the channel. So we're going to have a lot more positive outside the cell than inside the cell, and that causes the cell to be a lot more negatively charged because a lot of the positive ions are going to be outside the cell. So because we have that negative 70 membrane potential, that's resting membrane potential, when we have acetylcholine receptors that will open, allowing sodium to go down its concentration gradient and enter into the cell. So you can see here, we're going to enter into the cell because it's down its concentration gradient. That brings those positive ions into the cell, causing the interior of the cell to become more positive. So if we have this membrane potential, when we have positive ions coming into the cell, we're actually bringing up the charge of the cell. And now an action potential is actually fired when the cell reaches about negative 50 millivolts. So once we get a little bit positive because of those sodium ions entering the cell, we're going to fire an action potential once we reach negative 50. That's just a very simple overview about what happens when those sodium ions enter and why an action potential is fired when we have an increase of those positive ions entering the cell. We're going to go into a lot more detail about action potentials and what happens with those ions moving when we talk about the nervous system and action potentials moving along a nerve, but that's just a very brief overview of what happens when those positive ions enter the cell and why an action potential is triggered when we have that influx of sodium ions. So at step 3 here, we have that production of a muscle action potential because we have that inflow of sodium down its electrochemical gradient, and it makes the inside of the muscle more positively charged, and that change in the muscle potential. Potential is another way of saying like a voltage potential. It triggers a muscle action potential. So the muscle action potential will then propagate or move along the sarcolemma, and remember, down into these T tubules, and it causes the sarcoplasmic reticulum to release its calcium into the sarcoplasm. And remember, we did cover this a little while ago, but that the muscle fiber contracts by the sliding filament model that we talked about prior. So we have that influx of calcium. The calcium will then bind to troponin, which changes the conformation, it changes shape, and it moves that tropomyosin away from the myosin binding sites on actin. And then we have contraction, the power strokes that happen. Remember, we have those cross bridges that are made. And then calcium actually is transported back into the SR so that we don't have this continuous contraction because we have lots of calcium around a cell. We actually want to put that calcium back into the sarcoplasmic reticulum and restore that low level of calcium in the sarcoplasm so that we can actually allow the muscle to relax. Now, back here at the neuromuscular junction, the effect of acetylcholine binding lasts only briefly, and that's because acetylcholine is actually rapidly broken down by this enzyme called acetylcholine esterase. Remember when I talked about we need to break down that acetylcholine so that it's not continually activating those membrane proteins to allow that action potential to continue. We have to break down that acetylcholine, and so that we don't continue activating those receptors. If another impulse reaches this motor end plate here, another nerve impulse comes, we're going to have more acetylcholine released, and then the whole cycle is going to repeat again. Neuromuscular junctions are also usually near the midpoint of a skeletal muscle fiber, so you're going to have semi-equally distance on either side of a muscle from a neuromuscular junction. So the muscle action potentials that come from the neuromuscular junction actually propagate towards both ends of the fiber, and this allows for an almost simultaneous activation of all parts of the muscle fiber. So we're going to have an almost simultaneous contraction of the entire muscle fiber at once. Okay, so here we have a playable animation, which will actually show you all of the events that happen at the neuromuscular junction in animation form. So you can click on that link in your slides and you can check that out. And that's going to be the end of Chapter 10, Part 4.